good news and bad news on the exam. I really was trying to make good to my promise, and the good news is I kind of sort of did, and I'll explain. I thought I had all the exams done at about early this morning, and I was light some, and what happened is I had taken some exams to my other class while they were taking the exam, and I was correcting those, and I left them at school. So I have almost everyone's exam done. If you are really, so I won't be able to go through the answers with you yet today. I'll do that next week. But if you want to stop by after, give me till like 12 or something, like open lab time. You want to look at your exam, I'll get the scores up for you. So in a way, part of it is done. On the other hand, I was hoping to go through everything with you today. We'll have to take a rain check on that, but we can do that next week. So at least by the end of the day, you'll have an idea what your score is, which is often a very important thing for a lot of people. So that's that. Just way too many exams in my life right now. This is the new, the so-called new view of the Tree of Life, and we're not going to go in here and try to read it or anything, but this was in Nature in 2015, last year. It came out right before, a few months, I think it was in, June, right? It came out in June, before I went to Brazil, and people were very excited about actually the conference we had in Brazil on limnology in South America. Several people were on this voyage to do the work that resulted in this. I just wanted to start out today showing you that as much as we're learning, seems like there's a lot more out there. Kind of hard to read because I made this into like the banner for last year's course, but I can get you a better one and tell you this if you're more interested. But here's what let's see here. Yeah, here's what we're exploring with the bacteria. So our 30 to 39 different phyla are kind of like the big red things in this part of the tree. And here's the archaea. And even though we're going to keep it fairly simple, we'll go through the three main subparts of the domain in that. And we're over here in this little Tree of Life. We're slightly more related to the archaea than the bacteria, but the eukaryotes, people, dogs, trees, and it's too pixelated, and lizards. Okay, so what's interesting about this are these two groups. One that seems to be a whole nother kind of, what is it, a subdomain that's suggesting a whole new domain close to the archaea, and then over here on the bacteria. Now, to some extent you're like, oh, so what? Don't make me know anything else. I have enough to think about. And I'm not going to. I just want to say that the world around us is changing. We live in this wonderfully integrated and internet access world with all this information, and yet whole new domains of life exist, and you go, well, they're microbes, they're not a big deal, but you reflect back on maybe some of this genetic information, that the way that these things deal with life may be a whole different way of opening up other pathways. I mean, look what microbiology has given us, even in a broad perspective as we look through the last, back to the 1600s or whatever. So, just a little bit about the look ahead. I have not followed up since on this. I've been pretty busy doing other things, but if this is one of the things you want to take a look at more, we could at some point. So, okay, where we are at, and things that are more pertinent to our course, where we left things, we're saying, boy, these proteobacteria, there's sure a lot of them, aren't there? Yeah. So if nothing else, if you are thinking, boy, what's the most diverse group, you'll see that the proteobacteria, if nothing else, as you can tell, we're kind of skipping through this phylum, and still there's a lot of things. So we will finish proteobacteria today and get on to the Firmicutes and other ones. We stopped at the Pseudomonas, and the Pseudomonas are it's kind of confusing, because I keep saying, well, they're like the Burkholderia, and it really probably doesn't mean a lot to people who don't study the bacteria. The point is, both Burkholderia and Pseudomonas are closely related in what they do, in their phenotypic expression of things. They have a lot of genetic information. Okay, well, let's leave, kind of cross the road looking at Burkholderia. Let's focus on Pseudomonas. Pseudomonas, as we ended last time, Tuesday, was that we said, well, they're very metabolically diverse. They have a lot of genes, and with more genes, you have more options. And earlier on, we saw a graph where we showed as genome increases, we have more genes to express, and about the ones that go into transcription and translation stay about the same, but some of these other metabolic genes are kind of going up on this curve. You can look back in the chapters before and see that. Some say that and I don't know if this is controversial yet, but they report that in the Pseudomonas there may be up to 3 chromosomes. Okay, we have always learned, what have we learned? Well, bacteria divide by binary fission. Bacteria have 3 shapes, so we know that part of it's kind of out. We know binary fission is kind of cool, but that's kind of out the door too, because we know about spores and budding now with different ones. And that bacteria have 3 shapes, and they have a circular chromosome. 8 circular chromosomes, kind of what we are told early in our careers. And when I teach general bio, I say that too. It's the same thing when you start learning about matter and chemistry and all kinds of things. You learn general things, and as you go into advanced, you realize, oh, some of that stuff was pretty much true, but not all the way. So several chromosomes. We'll find that some bacteria even have linear chromosomes, or our chromosomes are considered to be that way also, linear. So diversity because of a larger genome. One of the things that these things do best, because they're oxidase positive, it helps us to reinforce the idea that they just love oxygen. Because of that, they produce oxidases, besides the cytochrome system. So they're really good at breaking down stuff. I don't care that you know about Brot and all this kind. This is an example. But if you want to break down oil, especially aliphatics and simple, aliphatics, they don't do so well in the aromatics. If you want to break down toxins, these are ones that you usually will find. One of my early papers, when we looked at the degradation of materials at the Savannah River site, we're very interested. Does anyone know about the Savannah River site? Probably very famous. The Savannah River site is where our country produced most of the nuclear material for the Cold War. So, does anyone know what the Cold War is? Okay, what was that? You just raised your hand, didn't you? You were thinking I wasn't going to call your bluff. What was the Cold War?
Wasn't it like a...
It's where we all put on big parkas, right? It was before global warming.
Something about the Soviet Union and the U.S. kind of going
Yeah,
at each
something.
other.
We had nuclear warheads. We were going to blow the crap out of each other at any moment, okay? And growing up in that era, it was a big mind thing. We just all... You either didn't take it seriously at all. If you really thought about it, it really freaked you out because you could become vapor at any moment. Well, we had to make nuclear material somewhere, so the Savannah River site in Georgia was it, and I was very interested in my second postdoc in finding out what happened to the microbes there and all the ammunitions and munition dumps and all the waste and so forth. So went down there and checked it out. Well, getting back to it, Pseudomonas putida was one of the things that we did hybridizations to kind of look at their relative numbers and try to figure out what they're doing. They break down stuff a lot. There's some pretty good correlations. Probably my third best cited paper is probably one on the Savannah River and pollution, but it was a few years ago, of course. Okay, so, what, like kind of the way I tell people to think about Pseudomonas, of the most diversity of bacteria, the most diverse, groups of organisms on Earth. The proteobacteria, these gram-negatives are probably the most diverse of all the bacteria. And within that, the metabolic diversity in Pseudomonas, and to some extent Burkholderia, are probably the highest of that. We always like to think of things, well, what's the biggest, what's the smallest, that kind of thing. Okay. This should give you some, as you're composing that essay for the final, when I ask you again, why is there so much diversity amongst the bacteria, this may give you pause to think about how incredibly versatile the gram-negative body plan or cell plan is for bacteria. Okay, we'll get to the gram-positives next. And they do a lot of cool and nasty things, but when we really look at the proteobacteria, and they're all gram-negatives, you have to think, what is it about that body plan, or that's, I shouldn't say body, cell plan, what is it about that, Do you want me to talk about it, or do you want to say, I'll reiterate, I hate to use your time up over and over again, but I think is a good teaching moment. So, refresh your memory, you got, moving the way out, you got a cell membrane, okay, a cell membrane that can really only take up to about 600 Daltons size of something, a molecular weight of about 600. That's about the size of glucose, and we know there's a lot of things regulating, well, if you got a little bit of polarity or something, we either have to have a channel or a cassette or something moving stuff through. What's next as we move out in the cell membrane? If I'm going inside cell membrane, what's the next outer area?
Cell wall?
It'll be part of, I think you'd consider it part of the cell wall technically, but there's kind of this gel area in between there, right?
Oh, is it that like sort of open area? I can't remember the name of it.
Sounds like first half. Okay, periplasmic matrix.
Periplasmic.
Okay, now, and I said, well, when they first discovered this, they thought it was a space, because they did EM. Well, EM dehydrates everything. But in the periplasmic matrix, this is kind of like cytoplasm, okay? It doesn't have all the cool, great stuff of cytoplasm. There's no protein synthesis going on in there or anything like that. But it's an area where you have enzymes, endo being still within the cell, but external to the cell cytoplasm proper. You have endo and exo, you have endo, ecto, and exo enzymes in the gram-negatives, okay? So in this space or matrix, you have another area where you can have a degradation and materials going on, besides what was going on with the electron transport system, which it's kind of on the side there. As we move out now what do we have as more of a layer, a wall?
Peptidoglycan?
Yeah, so we got peptidoglycan kind of in there, and that's pretty thin, and we move out into the outer membrane. Okay, the inner layer of the outer membrane is much like the cell membrane, and then we get into this lipopolysaccharide, which we are realizing can cause disease, but also has this kind of sugar, fatty acid kind of component to it. Okay, what can happen in that sugar matrix on the outside of the outer membrane is that you can, to some extent, do control diffusion. Okay, ectoenzymes that are sitting still connected in there can start to degrade some of these bigger polymers. Okay, that's huge. Recall Tuesday we were talking about how substrate diversity leads to metabolic diversity, which leads to species diversity. So that's a big deal. Okay, then external to that, in a lot of the gram-negatives, you have a capsule or a glycocalyx. It really depends upon. Sometimes it's slime, depends on how compact it is. Okay, again, attachment controlling diffusion. Diffusion is one of the biggest unsung controllers of life, okay? For us, for us terrestrial, dealing with not drying out, and diffusion and osmosis was a big deal. So now if you can attach, that's one thing. So now you close the distance between the cell and the substrate. Even if you can't, you're able to pick up a substrate and have and put out exoenzymes, okay, and then try to dissolve things. The most famous one being alkaline phosphatase. You probably study that in your other classes. Alkaline phosphatase is an enzyme that liberates phosphorus from organic molecules, and bacteria will exude that enzyme, and to some extent that bacteria will get the benefit, and other bacteria can also. So the question always is it altruistic or is it not? So when you think about that, having maybe this little bit more of a leaky membrane, but different options for attachment, for different areas for digestion, for transport, and then having components of the cell wall that may or may not induce disease or sickness simply by the contact of an O-specific antigen in the outer membrane, you start to see that, wow, I can really see why there's a lot of diversity, and that's not even getting into the cell itself that may be doing things anaerobically, aerobically, photoautotrophically, photoheterotrophically. So, these are all kind of some of the reasons for the variation. Okay. I think several things you should look forward to in the near future is another question on culture enrichment, which I'm not convinced we still understand, but also on diversity. And if you're going to address diversity a little bit more than their small, high surface area to volume, all those things are important, but now, kind of synthesizing some of these different ideas. So, Pseudomonas is flagellated. Most of us see Pseudomonas this way from a gram-negative. Here's a nice, this is just false color from fluorescent microscopy. Not a lot of, a real lot of information value there. Okay. One that we talk about, we always bring out a few of them that do disease, because people need to know more about that. Here's the concept of opportunism. If I haven't talked about it, certainly want to. To some extent, I think all bacteria will be opportunistic given the chance. You put like I tell everyone, I don't care how you classify, we're always supposed to work up to safety levels 1 and 2 at UIS, but it doesn't mean that you can't get really sick on a biosafety level 1, okay? The right bacteria in the wrong place at a high amount of number can create sickness. Opportunism in disease and infection means that the opportunity presents itself because of conditions. We talk here about burn victims. We'll come back to the other ones here, but in having a burn, maybe I mentioned this in lab, it's hard for me to remember, but of course, I'm just going to point as though I had a burn here, so okay, I had a burn, okay, and sure, the burn wants to seal itself, but usually you have a lot of lymph, and then you also have destroyed tissues. So that's if you look at it, that's a great place. It's warm. It's still by oxygen for the Pseudomonas. It's not like gangrene that we'll talk about in a little bit. And so it can infect and really take advantage of that and just start like growing like a culture tube in that burn. And of course, more severely you're burned, the more you have to watch that kind of thing. So people are aware of that, but Pseudomonas will take advantage of that. Same thing in urinary tract. Flushing of the urinary tract just from daily urinating is a big deal. People have compromised kidneys or other factors that affect their urinary tract, or a shift in the community associated with that. You can have then opportunists. To some extent, when we think of Staph aureus and Strep pyogenes in our throat, they're taking opportunity of you being out of whack, your microbes being out of whack. It's a different type of thing. A lot of us found out in labs, we were getting all these yellow, big yellow streaks on the mannitol salt augers. Like, do I have Staph aureus? Do I have MRSA? I think, I don't think you probably have MRSA, but you probably have Staph aureus. I mean, you might have MRSA. I mean, I don't know where you've been, but the thing is, that stuff's there. Are you sick? Most of you don't too bad, so I mean, probably not. But that stuff's there, and could take an opportunity to give you a sore throat or something in the future. Okay, let's talk about cholera. Have we talked about cholera? We have? Do you want to talk about some more? I want to talk about some more. Okay, Vibrio cholera. Vibrio, the shape. The shape of Vibrio is like a comma. It may fall into the spiral category of your three shapes in the world, but in here, Vibrio is this little comma. Okay. Now, where you can pick up, this is one that definitely moves through the water, fecal, oral avenue of contamination, which also can take into extent shellfish. Shellfish are, well, they used to be called filter feeders, now what? They're suspension feeders, but still, they're picking up particles, and one thing that bacteria love to do in water is to attach to particles or to coat things, and so shellfish can be a problem. What do a lot of people do when they're eating shellfish? A lot of people don't cook them, so it's like a real direct route. Does it happen a lot? Are people aware of this? Does it happen less now? Yes, all these kinds of things, but these are some of the historical kinds of things to think about. I do not care that you memorize these numbers. My point is over 340,000 cases and deaths in the hemisphere in 1991. Cholera continues to show up. Okay, it continues to be not only an epidemic or a regional type of killer, but a pandemic. It moves across parts of the world, and I think I have a map here in a minute. Okay, why? Do we know about Vibrio? Absolutely. Can we culture Vibrio cholera? Yes, we can. What is the problem? Let me tell you about two of them. Rita Colwell, who is the head of the American Society for Microbiology, if you want a great role model, female or men or women students, Rita Colwell is an excellent person to look into. She studied cholera, and one of the people in our lab at Kent State went to go work with her. She noticed two things. She found that Vibrio cholera is viable but non-culturable. This was interesting. It could fall into a state where, in some cases, you could culture it, but then also it could do its Vibrio cholera death thing, and you would go and look for it, and you couldn't culture it. This is a strange event. This is one of the things we brought up with that whole Great Plate anomaly idea. Only 1% is culturable. How many are really viable, and yet we can't culture them? Or how many are really dead? So Colwell brought that up. The other thing she and other researchers noticed is that cholera all of a sudden seemed to go away, and then all of a sudden it comes back. Now, the filter water, either the culture or not culture, with molecular, they say, well, we can find it. It's in these places that we're looking. Okay. They found out also that it has an alternate host. Okay, that at certain times Vibrio cholera kind of crawls into little microcrustaceans, like you guys know, Daphnia, the water fleas, copepod, they crawl in there and hang out. Okay, I don't actually think it's really symbiotism. I think it's just a host, like schistosoma in snails kind of thing. So, where does cholera, most express itself? Equatorially, and with cultures that really don't have good water treatment plants due to either numbers of people, due to economics, all those kinds of things. Okay, so this was an opportunity. This is thinking outside the box, and I want to tell you this because I think Paula was very insightful. I listened to her tell this story, and I thought that's good thinking. So, if you're in a culture, it's not that we tend to think, oh, those cultures are backwards. No, sometimes you're just poor. Okay, sometimes it's just the way it is. But one thing is that no one's going to drop off a big water treatment plant out of the back of 747 for your town anytime soon. What can you do? And when you watch people, die from cholera, it's not pleasant. So she said, well, okay, we can't treat the water. Even chlorination is beyond, to do this on a sustained level for these amount of people. She started a program called filtering water through a sarong. Do you know what a sarong is? It's a fairly, now it's more popular. People actually, does anyone know what a sarong is? Am I saying it wrong? Yeah, it's basically a stretch of cotton material, and oftentimes, like in Thailand, they wax different parts and dye, and it can be very colorful and dressy, and yet it's of course functional. You need probably something to wear. So she said, well, most of these families can't chlorinate, they can't filter, they can't whatever. You get the idea. But they have. It'd be like in our country if we said, we're going to filter all our water through our T-shirts. Okay, I mean clean T-shirts. Okay, anyway, so that's about the filter material of it. Okay, so and you go like, well, what's that going to do? Vibrio's smaller than that. Well, Vibrio is in a little microcrustacean about the size of a flea. You could see that this method would capture flea-like things and let the water go through. Now, it wasn't and never was thought to be 100% effective. And I don't accurately remember the success, but I remember being something like 40% less cholera when people started doing this type of very simple thing. This is applying what who would care that Vibrio is in a microcrustacean? So what? Those crazy ecology, it's saving a bunch of people's lives. So I like to tell that story because perhaps some of you will look at these things. Well, let's think outside the box. Let's try looking at things a different way. Well, cholera. This is actually in Europe. The people just showing the effect of the Grim Reaper just wiping out people. Cholera is not a fun thing to talk about, but we will. It's part of our job here. It is sudden. When it gives out the toxin, it has an effect on the body that basically just opens up the pipe, okay? And you start losing water. I think it's something like a liter or two an hour. I probably have it up here. 20 liters per day. I mean, you can't sustain that. What happens? Well, of course, massive diarrhea. Diarrhea to the point that even if your bowels try to make stool, they come out calling them rice stools, or basically just flushing stuff out of your body. Okay, what happens next? You become cyanotic. The venous portion of your bloodstream becomes more transparent, and you look kind of bluish. Okay, that kind of that seasick thing you ever see people get, like it's a real thing. This is that way. Your eyes sink into your head. Eventually, if untreated, you go into shock, and if you can't treat it fast enough, still you go into well, you die. So, can you treat it? Yeah, you know, in this country, fluids, the problem with trying to administer antibiotics is, they will kill cholera, it's just is that it's moving so fast that sometimes it's hard for it to, for the treatment to catch up to it. But, yeah, you're in better shape in the modern hospitals and stuff that we have, here, because, you know, we have more tricks. I think I covered most of that, and there, so. Yeah, the interior, so we talked about the different toxins. The one that operates on your intestines, basically, just basically pulls stuff off of you. Yes, it's not hard. I mean, we have antibiotics. It's just like the E. coli H57 strain. You know, just sometimes it acts so fast. You know, and you know now about growth curves and that kind of stuff. Like, oh yeah. Boy, it doesn't take much to have billions of things growing in the, yeah, stomach acidity helps, but the problem is that you're blowing so much fluid out of your body, it's hard to, for anything to really help. Yeah, purified water, I mean, I told that story. On the idea of water, we'll talk about John Snow. Do we know John Snow from other classes? I won't take up your time. Okay, let's talk about John Snow for a minute, okay? Mid-1800s, London, okay? John Snow is thought of as being like the first epidemiologist, as I have listed there. What is epidemiology? Looking at epidemic. What's an epidemic? That's kind of a regional outbreak of disease, as opposed to an outbreak, which is, it might be like Springfield could have an outbreak, but if Central Illinois got a problem, West Nile disease to a great degree, that'd be like more of an epidemic. You get the idea. So Snow is an aware man at this time, and he's noticing that the people of London are succumbing to disease, cholera, and he starts to map this out. He starts saying, well, where are there deaths? And again, this is an actual map, but the point of it is as he looks around London, which is nowhere as large as it is today, he's starting to realize that where he's seeing deaths happen to be around, his theory was certain water sources. Okay, now you gotta remember, now we know cholera moves through water and contamination and so forth, and yeah, I think 1800s London. Did they have sewage treatment or sewers then? I know the Romans brought them, but that's what, 500 AD. Were they working in London by that time? Because you know, in Shakespeare's day, when was Shakespeare? I mean, they're still throwing the chamber pots out into the street and stuff. I mean, you had to walk around and hope that you didn't get hit with someone's last night's going aways. Oh, so 1800, they're doing pretty good. So, okay, so we have certain stuff, but still, we have some kind of contamination of the water source. So he basically, goes to one of the squares where people are getting their water. It didn't have central tap water like we do. And he took the handle. Now, I don't know if London only had like a few handles, why someone else should go, well, hey, I got a handle, I'll just put it back on the pump. Anyway, it's one of those questions I've always wondered about. But for some reason then, this handle missing made people go to other water sources, and the incidence of disease in that sector went down. Okay. Now we kind of laugh, yeah, of course, cause and effect, it was cholera, it was in the water. Right, but, trying to look at distribution and how it's manifesting itself amongst population was a hard thing, like Typhoid Mary, trying to figure this stuff out. So, fairly clever. I guess, I've not been to London, have any of you? Well, if you get to London, I guess the pump is still there, and as I said, if you drink, what is it, Watney's Ale at the John Snow pub, that was I think they've cleaned it up. I don't hear people dying from drinking Watney's Ale, but I guess it's historically still there, so. Okay, epsilon, yeah, delta and epsilon. Like I say, this is just a way of organizing. I'm not gonna hold our feet to the fire. If I ever asked you about the subcategories, it would probably be from a table or something. I gotta look these subcategories up also. I can remember phyla, but, you know, Panicum always asks me, well, is that an epsilon or is it a gamma? Coli I can kind of remember, but anyway, so in the delta, what have we talked about? I think we mentioned some of these already. Desulfovibrio, Geobacter, Delvibrio, Desulfovibrio, and so forth. So we've already covered those. This is kind of a place setter for that. I think the delta and the epsilon, well, the delta are kind of interesting. You know, we're getting at the end of this diversity of possibly the most diverse phyla with, you can argue when you look at the phylogenetic tree that it's probably some of the most evolved of the bacteria if you're working your way back to the root where Thermatoga and the hyperthermophiles are. So, and then we start to see in here some fairly sophisticated aspects, haven't we? You know, we see predation. Okay, this is kind of interesting, but we see differentiation. Okay, some people are going and looking for that in class. We've talked about syntrophic relationships where no one organism can do the full reaction. You have to work with someone else. So, and then really crazy things like bacteria that can produce electricity. How would they have known, you know, when they evolved a billion years ago that they need to make electricity to make lawnmowers and light bulbs work? Now that's insightful. So they may have be able to have premonitions. Okay, epsilon. A few ones we want to talk about in here. Helicobacter, a real famous one. I think we've talked about this. This is the guy that drank the Helicobacter because he felt bad if he was to give it to his graduate students. You know, I talked about this story, right? I'll tell you the story anyway, real quick. Helicobacter, ulcers, right? Ulcers is an erosion in generally the stomach lining. If you erode the mucosa of the stomach lining, your stomach has acid, as you know, and it starts to go through the villi, where things are absorbed, and through the smooth muscle, which most your organs are wrapped in, and you get a burning into your stomach. That's an ulcer. It's irritating. You can't eat well. When I was growing up, my great aunts had ulcers from hard lives, making it through the Depression. I think would have given anyone an ulcer. And I remember them saying, oh yes, you know, Aunt Tilly has an ulcer. She can have milk and bread, and you know, has to have a very bland diet. And that's still a good thing. But we thought it was due to stress, and we thought it was a physiological response, you know, to humans. I have no idea if other animals get ulcers or not. I would guess if anything would, my cat would. Anyway, a few years ago, they decided to see, well, one guy had the idea that Helicobacter pylori may be the cause of ulcers, okay? And this is just showing that kind of burning in the mucosa. I think I've told you the only autopsy I ever helped with was someone who had succumbed to a severe ulcer, which got me off the list of wanting to do that activity again. But so you have, most of us have Helicobacter colonized. Most of us don't have ulcers, but you could. It's still being decided what kind of instigates them to start eroding things. But the big breakthrough, this guy got a Nobel Prize, is he started to think there was a cause and effect. He grew up a bunch of Helicobacter pylori in a flask, and he said, well, I got to test this. I don't know if people, you know, if it was they reported it as a joke. people often accuse professors of being abusive to their graduate students, but he decided not to give it his graduate students. He drank it, got an ulcer, and cured himself, and got a Nobel Prize for doing that. I guess I might give myself an ulcer for a Nobel Prize, I guess, but I don't know. I would just give myself an ulcer for trying to correct tests and get them done and done. All right, names, this is okay, don't worry about that. There's more to that, so in the delta and the epsilon, those genera that we covered in the functional chapter, I don't want to take up your time doing that again, but there are really a whole bunch of cool ones, right? Just make those connections when you're going through this part of that. Here's the zeta proteobacteria, zeta. We saw that in one of the opening graphs where it said, well, there's no culturable ones. There are some. We have some described species. I'm just putting this up for edification. Pay attention to the other subgroups. But just like we started the lecture saying, well, there's stuff we're still discovering. We never talked about zeta bacteria, 5 years ago. So now we have some iron oxidizing neutrophilic chemolithotrophic bacteria. Ouch! Okay, now, are there other iron oxidizing neutrophilic, okay, neutral pH, that's, we can decode all of these big words. Are there other ones? Sure. Why don't they slam them in those groups? Well, again, this is the frustrating thing. What things do functionally oftentimes don't line up with this 16S. So when I asked you to think about the limitations of the 16S classification, that's kind of one of them. We can line things up. It does give us order, it does give us ideas. It sometimes, though, if the bacteria, lining up function doesn't always work. So anyway, on the horizon, maybe when your kids are taking this class, there'll be other Greek names in front of there, more I'm sure there will be. We're finding a lot of stuff. Believe it or not, we are done with the proteobacteria. Get out of the way. Okay, just as that file featured gram-negative bacteria, these groups, which are, remember that some of those are those big trapezoids we saw early in this discussion about diversity. Firmicutes is one of those big trapezoids, and also actinobacteria. Remember the big four Bacteroidetes, Proteobacteria, Firmicutes, and Actinobacteria. So in here we have different subgroups. Some names we recognize, and already in our minds we're going, well, I know a little bit about that. Those are gram-positives. Yeah. So when you look at the phyla, there's no gram-positive phyla. You'd think there would be. Okay, but there isn't. We moved past that. You'll see the classification in the Berkey's manual, but when we get into phylogenetics as we now describe it, we have these kinds of words. Okay, they pulled out the Tenurichides out of the Firmicutes because these are the mycoplasma. The mycoplasma are the distinction there, right? The smallest of the bacteria. And why are they small? They have no cell wall. People are like, well, wait a minute. How can they be a gram-positive and they don't have that peptidoglycan thing you may be hearing about? Again, here's a case where by the phylogenetic comparisons, they fall in this group. If you were to really say mycoplasma, just like you might say acid fast, they don't quite, they go together, or they be separated in different ways. So there we have it. Corynebacteria, propionic acid bacteria. Yes, we'll talk about making cheese. Actinobacteria, the mycobacteria. What do we remember? Anyone remember anything about this genus? Yeah. tuberculosis. Okay, and then streptomyces, anything on that? Anyone have anything? Streptomyces is the group that produces the natural antibiotics. Okay, streptomyces also is the group that this time of year we talked about, they produce the geosmins. Remember that? Kind of that humic smell when you smell fresh rotting leaves. Old rotting anything doesn't smell too good, but in the fall now the leaves are falling. Oh, it's kind of moist and earthy smelling. That earthy smell is geosmins. So next week at Thanksgiving, like, oh, I love this type of year, the fresh turkey at our table, the family together, and the fresh smell of geosmins in the air. And I'm like, my daughter is learning so much at that school. They'll just love you, okay? So, what are geosmins? Well, let me tell you. They're from the group that produces the antibiotics. How interesting, how smart you are. Yes, you are. Okay, not going to talk too much about lactobacillus, but in this group of the lactobacilliales, okay, this subgroup that we're looking at now is the lactobacillus. Well, if you're in lab, we just did yogurt. That's one place we see it. Streptococcus. I don't think anyone in this room should have any problem thinking about where there's streptococcus and realizing there's good strep and bad strep. Where could you find about 30 species of streptococcus at this moment? Back to the throat, in your mouth. Your mouth is just full of strep, and of course it'll move there too. Strep next to staph in your upper and outer body, it's just filled with that. Whereas when we get into the intestines, those are the decomposers and the archaea and the proteobacteria more so. That's just a little factoid there. Okay, lactobacillus rod-shaped grow in chains. We've always done a strep grows in chains, but the chain thing isn't a mystery. It just means that as they grow, they divide at the ends, kind of like how we learn binary fission. You always see binary fission is bacteria, looks like this. Okay, great. The circular chromosome. Okay, and it gets a septum and it becomes this. Okay, we kind of see that, but we know, and those people who look through a microscope now, we also realize it's not quite like that, maybe. It's more so we have this, and then we have that, because we have to weave together, the Z ring and all that stuff of the peptidoglycan and so forth. And then we finally get to bacteria, and a lot of us have noticed that when the bacteria first divide, they are more coccus looking, and then they become more rod-shaped in general. So that's it. I like to bring this up now because when we look into things like micrococcus and Sarcina, we'll be seeing that bacteria, the way we get these other arrangements, is dividing on different planes. Okay, not hard to imagine. You'll get it right away. But okay, dairy products, we talked about that. And here we are. Okay, so there's lactobacillus, kind of get the septa in there. Nice long bacteria. Did anyone see any of this in yogurt? Anything that looked like that? I know most of you saw strep. Anyone? I didn't have a chance to see everybody's. Well, always take another look at it. You can show pictures of that at Thanksgiving too. You're eating that. Okay. Lactobacillus, a few things to say about this. Active anaerobe, sure, it does fermentation. We talked about it being in yogurt, also part of the vaginal normal flora, the fact that it controls pH is part of the natural flora that keeps that area healthy. Okay, and that's about what we need to know on that. We've covered homofermentation and heterofermentation before. So Streptococcus, very famous Firmicutes in this subgroup. We know this grows in chains, same kind of thing. Do the same thing, only with circles. Well, they're actually spheres, and you get that little chain thing, okay? Staph won't do that. Staph is more liberal in where it divides, okay? Some are pathogenic, oh my, yes, Streptococcus pyogenes, beta hemolysis, does it to your throat? Yeah, if you have that species of strep, and you look in the back of your throat, it looks like someone took an ice pick to it. I mean, and it probably feels about the same way. It really messes you up. But parts of it are completely okay with us. I know some of you are interested in Enterococcus. I'm not going to emphasize it here, but if you are, it falls into this group also. You see some enterics that are not gram-positive, was my point in including that. Streptococcus, a few things, string of beads, we know it's gram-positive, all these will be, catalase negative. In lab, I try to emphasize it's catalase negative simply because it's kind of indifferent to oxygen. It's not, it will do fermentation, but to tell you the truth, it doesn't care if oxygen's there or not because they'll go through glycolysis. If it's not going through fermentation, get energy that way, or go into fermentation and produce, recycle NADH and so forth. So, I just explained that. So, this wonderful picture here, we've talked about that, is where else can we find it? Well, strep's in buttermilk, which is kind of fermented milk products. You're right, in the throat, in our mouth. The thing about dental caries is the extreme growth of Streptococcus. Okay, we will cover, if we have enough time, biofilms on your mouth. Okay, but in case we don't, I'll take a second to talk to you about it. It's not on a slide, so if you don't remember, write notes, because the theme here is biofilm and also succession. Okay, now, what's a biofilm? Have I talked about it, or do you know? I don't want to start talking about something going, well, what the heck's he talking about? Any ideas? Okay, a biofilm, like it sounds, it's a film made of biological material. So that, okay, but come on, that's the obvious thing. So you can think of a biofilm as microorganisms embedded in a mucopolysaccharide matrix. Whoa, what was that again? Bacteria embedded in a mucopolysaccharide matrix. Oh wait, I know what bacteria are. What's this muco stuff? Okay, the mucopolysaccharide matrix is what you could say about the capsule or about the slime. Okay, when you have those long strain sugars emanating out from gram negatives, these guys don't do it as much, but they'll embed in others mucopolysaccharides. They're sugars, so they're acid mucopolysaccharide. You know what a polysaccharide is? Mucus just means the density of it. So you have this dense, slimy capsule, and you have bacteria embedded on that. Now, that's what a biofilm is. You have a recurring biofilm on your teeth that most of you don't enjoy. You don't enjoy the taste of it, or you're worried about this. So most of us engage in an activity of what each morning, and probably several times a day for some folks? Brushing your teeth. Now, here is both the fallacy and the truth of brushing your teeth. When you brush your teeth, you do not remove all the biofilm. You would have, now maybe, and I have not checked this, I would love to get a group of students and use those bleach things. Those bleach strips, you know what I mean?
Oh, the whitening ones?
Whitening ones, yeah, because I think that would remove the bacteria on your teeth, so we could give like 5 of you guys bleach strips and 5 people don't brush your teeth for 5 days. We could compare the fluoride. I think that'd be great. Or better yet, we could put bleach strips on one and just tape on the other people's teeth and just pull the tape thing and culture it. Anybody want to do that? Oh, he would, okay. Come on over. Okay, I've been wanting to do that for years. Nobody ever wanted to do that. I thought, wow, if you look at biofilm on people's mouth, we'll just use tape. Anyway, so on your teeth are a along with that mucopolysaccharide are also things that you know about. These glycoproteins, these different types of pili, of fibrae, okay? Not to the point of just like, okay, I'm gonna brush my teeth and scrape them off. They'll actually embed into the enamel. So, what is plant succession? If you had to do that, now I know you've learned this in general bio, and you probably learned it in grade school now because we're very environmentally aware. If I had to describe plant succession, we're connecting concepts now, biofilm, teeth, and succession. What is succession? Oh, come on. Start with a bare field. What grows in the bare field? If I plow a field or I have a rock or something, what is primary succession? You guys haven't read Henry Horn or Gleason or any? Go ahead, you had an idea. Yeah, you have an orderly change of species. Primary succession is, let's just say the glacier moves out, and you got bare rock, eventually it'll be dust. What's gonna grow on there? Lichens. Okay, now, given a little bit of time, you get a little bit more soil, you get moss, then you get plants, and then the shrubs come in, and then the bigger trees, and then the shade-intolerant trees live underneath the shade-intolerant trees, and you get canopy, and then you get a climax successional stage, which they argue doesn't really exist, but we all learn, well, most of us learn about it, and then it starts all over, okay, succession. I don't know how to look that up, okay, that's a concept I think you should know, and if you don't, look it up. I think I've explained it, but I think you want to just do the wiki on that at some point, because, plant succession. I'm trying to relate that now back to your teeth. If you, your tooth emerges as a child, it's probably fairly sterile, not for long, okay. On your teeth, you have bacteria. Usually there's a layer of strep that lays itself down. And then there's usually a secondary layer of colonizers behind it. So you have pioneer species, and you have species that come behind it. If you don't brush your teeth maybe for a day or two, and a lot of you, even if you do, a lot of us have actinomycetes. We haven't talked about them. Those are those long. If you do a gram stain on your teeth, they're the long, thin ones that kind of look like hyphae, but they're that. And we get more. We get fusiform bacteria, and we get more and more things. And if we don't grind back, we get this film. This is iodine on the biofilm. Okay, so they just stain this for our and then you get enzymes. You know that if you're fermenting sugars, you get acids. That's why I said don't eat so much candy. You get fermentation of sugar in your mouth. It erodes your enamel. So you do that, and you get it in here, and you start, eroding your mouth, and you look like some guy with bad teeth. So I'm sure I think the guy's saying Aquilon. He always had those bad. Anyway, so when you grind, so you wake up in the morning, you go, oh, see, my mouth tastes like a pigeon slept on last night. I gotta go to the bathroom and brush my teeth. You grind this away, and even you'll use Listerine, and contrary to what the commercials show that all the microbes shirk away and go back, Listerine will help to reduce that. It will not blow off the biofilm. Your toothbrush will do a good job, but unless you are wash your mouth on bleach or something, you're not really going to get rid of it. That's okay. You get it down to a level every day. You think, why do I have to brush my teeth every day? Because they're growing back that fast. Just like 1/3 of what's going out of you is back. They're growing really fast in you all the time. So caries can be a thing. Strep, though, is a big part of your mouth. You start out as a little baby. Your mouth is pretty much sterile. Within 14 days, I think you have something like 150 different strep on you. Why? Because your you're sucking on stuff, you know, you're sucking on everything, and strep loves your mouth. Just quickly, one way we can tell different types of strep and other bacteria, but I'm bringing it up now, is that if we put it on a blood plate. I realize in the lab we talked about, I just want to make sure everybody gets this. If we were to take a culture, this is a pure culture, we do a streak plate, and now we have colonies. Don't pay attention to this, it's too much. And we see this nice clearing, we go like, oh, that's a nice clearing. But what happened is that hemolysins of the type beta have been emitted by that type of strep, and it's thoroughly destroyed the red blood cells. This is blood agar. So we say, wow, watch out for those guys. Okay, strep pyogenes would be one of those ones that would do it to you. Okay, if it only partially oxidizes the hemoglobin, you know, the iron-carrying oxygen component of our blood, you know, kind of like I always say a bruise, that's the way I think of it, but anyway, then it would be alpha. So we can tell things by what these things emit from their tiny little bodies. Okay, very beautiful picture here where we have, you can see how there's been already fission there, and we have this nice connection where we're still intact, and now they're going through the next generation. Isn't that a great picture? So we see all that type of thing we talked about, the different rings and how they weave together the peptidoglycans there and so forth. We have enough. Okay, Listeria. Listeria, I just say, in the food industry, you say Listeria and it brings fear to people. Listeria outbreaks can be terrible, associated with lunch meats. Oscar Mayer, he was always worried about Listeria. Not that Oscar Mayer is bad, it just is that you're very concerned about, you know, like any big corporation not having outbreaks of things. I probably won't go too much into it. I do want to talk about Sarcina. Sarcina both as a genus, but as a shape, like Streptococcus being beads and Staphylococcus being clusters. Sarcina is a type of arrangement that they name, you know, they name the genus after that arrangement. So we'll talk about that. And staph. So yes, all gram-positive, catalase positive now, not catalase negative. That was strep. Okay? And Shane's obligate aerobes. Let's keep moving up. Let's move this way. Okay, let's feature staph. We all, I think, have an idea of staph. They like air. I mean, they'll do well with they like air. This gram-positive goes without saying. The way we can get staph, and it's really, we're quite lucky at this actually, that they can tolerate high amounts of osmotic pressure. So we use salt agar. I think salt agar is about 15% sodium chloride. You could use any ions you wanted. Staph will still grow on it. That's kind of interesting. Okay. Many species are pigmented, that is true. So Staphylococcus, now, I forget why I included this, so let's not talk about it. I just don't remember. But here, when staph is dividing, you see, now I know it's an electron micrograph and you see dehydration through here, but see, it's not dividing on that same plane, you know, of tip to tip. What's going to happen is one time it's going to divide here, you know, and instead of just continuing that division and then dividing that way and so forth, it'll have another division plane, you know, that'll be on a different portion. Okay, it's not as regimented as strep. So then, you know, then if you have a division plane that's maybe over here, you'll get something. But if this division plane is here, now you can kind of see that you get this cluster. I don't care that you describe that, but again, you know, I think people start to say, well, why do I see that stuff? You know, what's that all about? And that's his division plane. And maybe that's what I'm showing you. I just really can't remember on that one. I don't have a scale to help me. So we see these clusters, okay, as opposed to beads. Okay, I do want to talk about Micrococcus next, coming up. Really want to talk about Micrococcus is the division, but sticking with staph. Staph epidermidis, if you're in lab, you're probably sick of hearing about it, so I'll keep it short, but it's non-pigmented epidermidis. Think of epi, above your dermis, okay? It's on your skin. Is a good place to think of it, but staph epidermidis will show up in the throat. Now, they just came up with a study that we used to think our lungs were sterile. That's what, you know, if you read, even, I don't know what Brock says, but the nurses were reading their book and they still teach that the lungs are sterile. The lungs aren't sterile, they have a normal flora also. We find that staph is part of the normal flora, but when we start getting big pussy things in our lungs, we have pneumonia, and we have to be concerned about that. Strep, typically, staph epi typically is not a problem with it. This coagulase negative staphylococcus is one way of determining the degree of enzyme production. One thing that we looked at in lab is we compared DNase degradation. If your DNA is positive, you bring on DNase, generally your coagulase positive. So it's one way of distinguishing, you know, another way of just helping confirm what kind of staph you have. This is an old picture from ISU, but I like it. Staph epidermidis showing the clustering, but also helping to reinforce the idea that EM dehydrates. But for those of us who think that there really isn't much mucopolysaccharide with these organisms, there are. You know, when you look at that colony, it is a little drier. It's not as mucoid as the gram negatives, but there's some stuff there. So, aureus, okay, the other aureus, like the sun, yellow, generally will have a pigment. Sometimes some strains don't. We know where to find it. We've talked about that. Big deal, of course, now with MRSA. No longer news for us. Now it's the question of how bad will it get. I think on the last test, no, it wasn't your class. I mean, can we have mutants of mutants and superbugs giving way to superbugs? Well, yeah, to some extent, till you just start, outdoing what antibiotics will do. So it's possible. Talked about mannitol salt agar. Here's the big difference. I don't know if everybody saw the difference, so here it is in lecture. This is staph epidermidis on mannitol salt agar, so the phenol has no reaction, there's no acidic reaction. Here, with fermentation of mannitol, you have production of acid, and therefore it's an easy way of telling aureus from that. My phone sometimes just starts talking to me. I have to figure out how to turn it on. One thing we don't hear about too much anymore, but a good example of exotoxin. Now, Entero was the lumen, but this is a toxin going out, exo from the cell, was toxic shock syndrome. Okay, I have not heard of incidences of this. Maybe you guys are reading the news more than me, but I remember this kind of like in the 70s, 70s and 80s. And what was happening is that primarily women were developing fevers, rash, vomiting, diarrhea, and sometimes they would be dying from post-menstruation, okay, not menstruation, and so it turns out that the tampon material was capturing the blood, which it is designed to do, but it was poor absorbance, and so you were having secondary growth and rapid growth of staph aureus into it, and a release of an exotoxin that is in an area that can be absorbed into the host, the person. And so, depends upon what was happening, people got really sick. Well, one, people started talking about this more like, hey, this is happening. Manufacturers started to think about, well, let's rethink about what this stuff is made of. I don't even know that's I don't know, I mean, I have not heard that's a problem anymore, but, you know, it was one of those things where, you know, like the Gremlin, you know, making and the Pinto, making a car with a gas tank that explodes, maybe we should change the design of that, I think one of these things that people figured out. So, that's the exotoxin, just remind you what that is, we talked about that already, so you guys are sharp in the nose. Okay, I just want to talk about this genus briefly. In the Sarcina, we have these division planes that are very symmetrical. So I think I have another one, but a Sarcina, then, if you're dividing symmetrically on two planes instead of just end to end, you know, then you get these really nice boxes. A Sarcina is like a box of 16, because you divide like 4 times, and then, you know, and then before it moves into another cluster of its self. So, yeah, and I know this kind of looks like nuclei and stuff. It's just the way that they took these shots. When you get into this kind of magnification, you have to be real careful interpreting artifacts. But these are not eukaryotic cells. Micrococcus kind of does the same type of thing. You start with one cell, you have the division plane, looks like strep, okay? But then it divides on the other plane, it does that several times, and this is the limit of my artwork, but you get the idea that you get this cube of cells. So oftentimes, if it's not a fully developed 16, sometimes micrococcus is just called a packet. Okay. If it's very much like that, Sarcina. In advanced micro, we get mud, and we look at methanogens, and there's a enzyme that makes methane that you can fluoresce. So we light up mud with fluorescent, and we often find Sarcina. There's a methanosarcina. So, like so many of these things, Bacillus, okay, it's a genus, but it's a shape. You know, Sarcina, it's a genus, it's a shape. So you just have to, you know, pay attention to how people are talking about that. Okay, Bacillus and Clostridium. You feel like talking about it, or you feel like ending? I know this is a lot, so I thank you for your patience, but we have enough for today? Oh yeah, Clostridium is great because we have